Hello guys, in this video we will discuss Avery's transformation experiment. In last video, which is Griffith's experiment, we learned that R2 was transformed into S3 and he concluded that R2 has absorbed something from S3, which is why non-virulent R2 got converted into virulent S3. But guys, what was absorbed he wasn't able to find. So after him Avery with his colleagues MacLeod and McCarty around 16 years after, that is in 1944, they continued Griffith's experiment but in vitro pattern. In vitro means in test tube. We know Griffith performed experiment on mice, but this guys continued their experiment in test tube. So what they did is that they took S3 bacteria, heat killed it. So as now it is heat killed, it becomes non-virulent. So now with the help of detergent, they lysed the cell. That is, they break open that cell and extracted cell debris. Now the cell debris will have all lipids, carbohydrates, DNA, RNA, proteins all mixed. So first they remove lipids, carbohydrates with enzymes. So now it is only having DNA, RNA and proteins. And then in this first they added protease. Protease destroys protein. So from DNA, RNA and protein, after adding protease, protein is destroyed. So we only have DNA and RNA. And then they added in it live R2 strain in this mixture and found that transformation took place. That is, R2 got transformed into S3, live S3. So as conversion is happening, protein is not playing any role, as even though protein is absent, conversion is taking place. So protein is not the transforming agent. And then what they did is they in this DNA, RNA and protein mixture they added RNAs. Guys, RNAs destroys RNA. So now as RNAs has destroyed RNA, we only have protein and DNA. And then in it they added live R2 and found that conversion is again taking place. So as conversion of live R2 is happening to live S3, uh, even in absence of RNA, so RNA cannot be the transforming factor, right? Now next they added DNAs. DNAs destroys DNA. So now DNA is destroyed and we only have RNA and protein. So again they added live R2 and found that live R2 is not converted in S3. That is, when DNA was absent, there was no conversion of live R2 to live S3. So it is DNA only who is responsible for transformation. So this is how Avery, McCarty and MacLeod proved that DNA is the genetic material and not RNA and protein. So in 1944 we got to know that DNA is genetic material, and after 8 years of this experiment, that is 1952, Hershey and Chase by publishing research paper gave more evidence that DNA is the only genetic material.